Hello everyone, my name is Bo Li. I'm a postdoc from Dr. Kingfei Lab. Today, I'm glad to introduce one of my research work, zygotic activation of transposable elements during zebrafish early embryogenesis. Transposable elements, or TEs, can occupy large parts of eukaryotic genomes. For example, TE can account for 46% of human genome and 51% zebrafish genome. For the most of the time, TE are under strong genomic control and remain silent. However, under some specific scenarios, TE can be reactivated. For example, in the cancer cells or the aging brain cells, we can observe elevated TE activities. Surprisingly, TE can become very active during early embryo development. Prior studies based on the short-read RNA sequencing technology have already revealed that the T reactivation is a very conserved process in many mammal species and in zebrafish. Besides self-expression and the potential the following retrotransposition, T can also affect the gene transcription. For example, T can provide cis-regulatory elements for T expression, such as the promoters, enhancers, and terminators. T can also insert it into the gene body and create a gene exons. There are also many other approaches for TE to interact with the gene transcription. So far, short-read RNA sequencing technology have already made great achievement in T study. However, there are still some difficulties in accurately quantifying the T expression. The first problem is about the multiple mapping. Because there are so many highly identical T copies across the genome, when you align the short-read RNA-seq data, you will achieve a very high multiple mapping rate across the T region. Therefore, you cannot determine the exact location for this rate. The second problem is there are two major kind of a T transcript. The first is the autonomously expressed T. They are the real T transcript. The other one is the T gene chimera transcript. If this part of the show very highly similarity with this part, and the rates from this region can be wrongly aligned here, therefore affect their this T codification. Due to the problems here, so the previous studies based on the short-read RNA-seq data typically can find thousands or even tens of thousands of expressed T copies. However, we think the actual number should be much lower than this estimate. Compared with the short-read RNA sequencing technology, we think the long-read RNA-seq technology is an ideal solution for T identification and codification. With much longer read lengths, the long-read RNA-seq technology can minimize the multiple mapping rate and also can provide the fully coverage of T transcript. Currently, the long-read RNA sequencing technology have already successfully applied in T study in both the animal and plant species model. In this study, we want to address the following 5 questions related to the T activation during zebrafish early embryogenesis. Number 1, among the 3 million copies, we want to know which specific T copies are active. Number 2, we want to know what exactly time are these T copies activated. Are they activated simultaneously or at a different time point? Number 3, we want to where are these T transcript transported? Are they still localized within the nuclei or exported into the cytoplasm? Because they can lead to very different biological consequences. We also want to know how this T activation is regulated on the epigenetic level. Finally, we identified the two essential genes during zebrafish early development, and we studied their function. To do so, we established a T-derived transcript identification and classification pipeline, and we collect the total RNA from 11 development stage, and we did the standard Pacbell RNA sequencing, and we used the full-length transcript for the transcript identification and used both the full-length rate and the non-full-length rates for the codification. And then we classified all the full-length transcripts into the following three categories. The T-alone transcript, representing the autonomously expressed T transcript, and the T-gene chimera transcript, and the gene transcript, if there is no any T insertion. To further improve our T annotation, we add a step of manual curation, including merging the highly identical transcripts, adjusting the T lossy and the T transcript structures, and also removing the wrongly annotated signs. So this results in a final T-alone data size, including the 706 T-alone transcript from 550 T loci. So it represents a very diversity populations covering the 4 types, 30 families, and 210 subfamilies. So LTR transcripts are the most, following by the DNA transposons and the lines. We only identified 2 transcripts from sign class. This figure represents the details about the T classification. Among the 706 T-alone transcripts, most are generated from a single T locus. However, we identified 48 chimera T-alone transcript, which means this transcript generated from 2 and more neighboring T-alone loci, and they belong to the different T subfamilies. Here we show 2 examples to show how this chimera transcript generated. This is an example for the nested T insertion. Here we can see there is a DNA transposon inserted into a BEL LTR transcript and then generate together to form a chimera transcript. This is an example for the read-through transcription. Here is a Gypsy24 LTR retrotransposon, and this transposon lost its 3' LTR, where the transcription termination signals there. Therefore, the transcript occur here and continue to here, because there is no terminal signals. And then finally, they generate a very complicated chimeric T-alone transcript. With our high-quality T annotation, we can observe a different transcription patterns among the different T classes. For example, the DNA transposon prefer to have multiple isoform and multiple introns. In contrast, most of the line do not contain the introns. So LTR show a mixed pattern. The some of LTR have a single exon, but the other have multiple exons. With the full-length T-alone transcript, we continue to investigate whether or not these T-alone transcripts still have the conserved domains which are required for the T transposition. So what we find is the most DNA transposon and LTRs lost 1 or more conserved domains. Especially for the ERV transcript, we cannot identify any one ERV transcript having all the 6 conserved domains, suggesting that this kind of a transcript completely or partially lost their retrotransposition ability. However, for the line transcript, the most of the line transcript maintain the two conserved domain, suggesting that most of the line may still have the ability to jump around the genome. We directly compared the performance between long read and short reads in T-long transcript identification. We found long reads typically can identify thousands of expressed T copies. It's much higher than the long reads. And T-long transcript identified by long reads are much longer than those from the short reads. Importantly, the T-long loss identified by the short read data are not largely overlapped with those from the long read data, suggesting that a large amount of expressed T-long loss identified by the short read data may not be the true expressed copy, because the aligner will randomly report one location if they find the multiple equally best alignment. Here we show a typical arrow from the short read alignment. For the long read data, you can easily identify the full-length transcript for this T copy. However, for the short read, they annotate this region as 3 shorter transcripts. A short summary for the first section. Among 3 million T copies in zebrafish, we identify only a tiny proportion to be active. It's only 515 active copies. So we apply these active copies across the genome, and we find several clusters, especially on the chromosome 4. As we know, the chromosome 4 is a sex chromosome in zebrafish, which have a very low recombination rate. Maybe it can used to explain why these copies can survive and escape from the removal by the recombination. Zygotic gene activation have been well studied in zebrafish, including a minor wave at 6-4 cell stage and a major wave at 1K cell stage. To study zygotic T activation, we first need to separate zygotic T transcript from maternal T transcript. We define a transcript as maternal if having TPM larger than 0 at either the fertilized egg or 1 cell stage. We only identify 11 maternal T-long transcript, including 9 DNA transposon. Based on the number of zygotic T-long transcript at each stage, we can define 2 waves of ZTA. So minor wave at the 1K cell stage and the major wave at the oblong stage. Compared with the well-defined ZGA, both waves run much behind the ZGA. We also perform expression correlation analysis between 2 neighbor stages to confirm this analysis. We suppose that 2 stages with different expression composition will show low correlations. For both gene and T genes, we observe that the 2 lowest stage-by-stage correlation corresponding to the minor and major wave of ZGA. Therefore, we think the 2 lowest stage-by-stage correlation in T-long group should also reflect 2 waves of ZTA. As expected, the first lowest correlation occurs between the 6-4 cell to 1K cell stage, which is consistent with the minor ZTA. And the other lowest correlation occurs between the high and oblong stage, which is also consistent with the major ZTA we defined. We also select 10 T subfamilies for which experiment. Due to the lower resolution, the minor wave of ZTA cannot be recognized. by the fish. But the major ZTA can be confirmed, which take place after the oblong stage. A recent study on mouse embryonic development reported gene activation occurs as early as 4 hours after fertilization. However, MERVL retrotransposon are activated at 8 hours. These results may suggest that the delayed pattern of ZTA compared with the ZGA may be conserved between zebrafish and mammal. So we reanalyzed the data and found most TE subfamilies are activated after 12 hours. Only 6 subfamilies show activation between 6 to 12 hours post-fertilization. Previous studies suggest that the minor ZGA are critical for establishing the major ZGA. If you block the minor ZGA, you will observe expression reduction in major ZGA. These results inspire us to think if earlier ZGA is also very important for the following ZTA. In other words, it's possible that the ZTA requires some essential factor from the ZGA, so it needs to activate it after it. To find some evidence to support our hypothesis, we analyzed a dataset with some zebrafish mutants and embryos by treatment with transcription or translation inhibitors. In the original study, the authors found alpha-amnitin can block zygotic gene activation. What we find is alpha-amnitin can block not only the zygotic gene activation, but zygotic T activation. In a triple loss of function mutants with 3 important transcription factors, POU5F1, SOXB1, and NANOG, we also find the similar results. Both ZGA and ZTA can be affected by loss of these 3 pioneering TFs. We also find mRNA rescue can restore the ZGA and ZTA to the normal level. When blocking the translation of zygotic activation, the original study reported that the early zygotic gene activation is not affected, suggesting the maternal materials can fully support the early ZGA. However, the major wave of ZGA are dramatically affected. Our analysis showed that after blocking the translation of zygotic genes, we can observe a global reduction in ZTA, suggesting that ZTA very likely requires some critical proteins or other materials from ZGA. We further studied 2 zebrafish transcript factors, MXTS1 and MXTS2, which are 2 autolog genes to human DUX4 and mouse DUX. Human DUX4 and mouse DUX are well studied for their function in HERVL and MERVL activation. If you knock out mouse DUX, you can observe a significant decrease in MERVL expression. During mouse embryonic development, DUX is activated at 6 hours, and the DUX-responsive genes and TEs are activated later. For example, MERVL is activated at 8 hours, as we mentioned before. This suggests a sequential or step-by-step fashion in activation. We find MSTS1 and 2 can bend 5' LTR or promoter regions of 9 ERV subfamilies. Importantly, MSTS1 and 2 are strictly zygotic genes, are activated as early as during 64 cells to 1K cell stage, preceding the defined ZTA, which is from 1K cell to oblong. After blocking the translation of MXTS1 2 and other zygotic gene. We observed all 21 ERV subfamilies show expression decrease compared with wild type. We then continue to characterize the ZTA landscape on different levels. We find the DNA transposon a major form of T-along transcript before ZTA. After ZTA, LTRs, especially the ERV, rapidly dominate the T expression. As previously reported, there are extensive variation in activation and expression pattern among T subfamily and families. With long-read RNA-seq data, for the first time, we can explore the expression heterogeneity of TEs on lockers and transcript level. And because of similar results between the lockers and transcript, here we only show the transcript level analysis. 695 zygotic T-along transcript can be divided into 8 clusters based on the expression pattern. We calculate the entropy for each subfamily, which is used to measure the complexity of expression within T subfamilies. We can observe that some subfamilies with lower entropy show simple expression pattern, such as the Gypsy 117 here. Some subfamilies with higher entropy show a much more complex expression pattern, such as this BHIK subfamily. In summary, most T subfamilies show more than 2 different activation time and more than 2 expression patterns. The expression of this BIK subfamily has been studied before, and its promoter has been cloned and validated for specific expression. Our VISH experiment also confirmed that it is specifically expressed in Mesododom, but other 2 close subfamilies are not. We identified 19 expressed loci for BIK subfamily, which show diverse expression pattern. However, when we analyze their promoter regions, we observe high identical regulatory sequences among them, including the CCAAT and the Tata box motifs, suggesting that the expression heterogeneity among highly identical T copies may be regulated epigenetically. We further studied the relationship between evolutionary age and T activation in 3 independent approaches. First, we calculated the cumulative divergence value for each T copy and found active T copies show significantly lower KD values, suggesting evolutionary younger than silenced copies. Second, we conducted a de novo approach to discover intact LTR and calculate their insertion years by comparing the sequence divergence between 2 LTRs. Then we separate them into silent and active group. KS test shows that active groups show significantly higher proportion of LTRs with young insertion age. We also performed a comparative analysis with zebrafish TU reference and other 3 genome assembly from different zebrafish strains, including AB, Nadia, and CB. We extracted 25 kb upstream and downstream of each active T-along lockers, and blast this sequence against the AB, Nadia, and CB genome respectively. This more than 15 kb sequence can guarantee us to find the real autologous regions. After that, we can infer the presence and absence of active T-along lockers in other genomes. Here we show 3 examples. This one is this TE is present and at AB genome and Nadia genome and CB genome. And this TE is present at AB and Nadia, but absent from CB, and this TE present and AB but absent from Nadia and CB. Based on a previous established evolutionary pedigree among these 4 zebrafish strains, we can summarize that there are 335 TE as TU specific, and 172 is shared by AB and TU, and 30 shared by Nadia, AB, and TU, and 14 shared by all 4. From TU specific to shared by all 4 are from evolutionary young to the old. We then tested the relationship between evolutionary age and expression pattern. We first test the cumulative divergence and T abundance, and we do not find a linear correlation between them. We then classified TO specific and TOAB shared TEs into evolutionary young group and others into evolutionary old group. We can find a distinct expression patterns between them. Evolutionarily old groups show low expression, and the evolutionary young groups tend to show higher expression, especially after the major wave of ZGA. We also established a long-read-based pipeline to study allele-specific expression of TE. In our workflow, there are 3 major steps. The first step is we align the CCS rates onto the genome and recorded the mismatches between rates and genome. Second, we map the whole genome sequencing data from 2 parental samples onto genomes and perform SNP calling and genotyping. Third, we compare the mismatches and SNP datasets to infer the origin of race. For example, these races have AG mismatch, which are consistent with the maternal SNP here, AAGG. So this race from maternal genome. For some races, we do not have enough information to infer the parental origin, so we discard this kind of race. Here is an example to show how well our pipeline can separate CCS race into maternal and paternal group. Here we only use 1 informative SNP. Then we can successfully separate all the F1 CCS race into the maternal and the paternal group. Based on this pipeline, we can study the allele-specific expression of TE at each developmental stage from 1K cell to shell. So from the high to shell stage, we can identify the maternal specific expression TE, paternal specific expressed TE, and allele specific expression TE, which means the maternal and the paternal show twofold differences in expression. Interestingly, when we look at these T loss across all development stages, we find 9 maternal and 4 paternal Ts showing monoallelic expression during the entire ZTA. We hypothesize that there are 2 major approaches to cause this monoallelic expression: T polymorphism or the gene imprinting. The T polymorphism means that this TE lockers is only present in one genome but absent from the other. If these T lockers exist in both alleles, the monoallelic expression may be regulated by the similar mechanism as the gene imprinting. To test this hypothesis, we established an approach based on the whole genome sequencing data to test the presence and absence of T loci in both maternal and paternal genomes. If their sequencing rates can cover both the TE and the flanking genomic regions, we can determine it exists in a certain genome. There are two examples to show that T polymorphism can lead to monoallelic expression. Finally, we identify 6 cases of monoallelic expression, which may be regulated by imprinting, because the presence of these TEs can be validated by whole genome sequencing data. A short summary for the second section. We defined 2 waves of ZTA, which are initiated after 2 waves of ZGA. The delayed ZTA is likely because ZTA need necessary materials from earlier zygotic gene activation. We characterize the ZTA on different levels and reveal that ZTA is a complex and heterogeneous biological process. After showing the time course landscape of ZTA, we continue to study the spatial characters of ZTA. We investigate if there are any difference in subcellular localization of T-alone transcript and gene transcript. We developed a method to calculate the relative NC ratio because RS-seq data cannot be directly used to compare the expression level between nuclei and the cytoplasm. And to test the performance, we first calculate the relative NC ratios for protein coding genes and link RNAs. Because previous knowledge indicates that protein coding genes tend to localize in the cytoplasm, whereas the link RNA tend to localize inside the nuclei. As expected, we can observe the higher relative NC ratio for the link RNA, suggesting the nuclear pairs for the link RNAs. Then we calculate the relative NC ratio for the T alone, T gene, and gene transcript. At 3 deep development stages, we can observe the T alone show the highest relative NC ratios, suggesting the most bias to the nuclear subcellular localization. We observe a positive correlation between T fractions and the relative NC ratio, suggesting that the T sequence may have potential functions in regulating subcellular localization on mRNAs. At the T subfamily level, DNA transposons show lower relative NC ratios than LTRs and lines. We selected several T subfamily for fish experiment. We find DNA transposon prefer to localize within cytoplasm. Lines are strongly nuclear localized. LTR shows stronger signal inside the nucleus than the cytoplasm, although the signals can be detected in both locations. This fish result is consistent with the estimated relative NC ratios. Here, the red dots represent the corresponding NC ratios for these 3 T subfamilies. A short summary for this section. We find T-alone transcripts show higher preference to localize inside nuclei compared to gene transcripts, especially for lines. Our results may shine a light on the paradox between the widespread T activation and the rare T transposition. In our data, DNA transposon and LTRs can be relatively efficiently exported into cytoplasm. But because lacking conserved domains, no protein necessary for transposition can be synthesized. In contrast, for lines, which can encode the protein for transposition, however, cells strongly control its transport into cytoplasm, therefore block is translation. Through these selective mechanisms, cell can minimize the T transposition rate and protect the genome integrity. Next, we investigate epigenetic regulation on TE. We first take a look at the chromatin accessibility. Across T loci, we observe the chromatin accessibility increased from 256 cell to dump stage, and then started to decrease. However, across the G loci, the chromatin accessibility continuously increased until 80% apibody stage. We also checked the H3K9ME3 histomodification, which is the major repressive marker for TE. We observed increasing deposit from 6-4 cell to shell stage, indicating an elevated repression control on T activation. However, genes do not show H3K9ME3 increase during the same period. Previous epigenetic profile suggested that TE activity may be strongly controlled after shell stage. Because we do not have long-read sequencing data covering stage after shell, we analyze a short-read RNA-seq dataset. Despite no locus or transcript level analysis, we can still use it to describe the overall pattern. As expected, the expression pattern before shelled are very similar between short-read and long-reads. After shelled, the short-read data show 75% apibody is the expression peak for T activation. After that, T expression starts to decrease and finally maintain a very low level. Based on that, we can divide the zebrafish T activity during early development into 3 phases: silence, activation, and repression, which is also consistent with the epigenetic profile we studied here. Previous studies suggest that PV pioneering pathway is very important in controlling T activity in both male and female germ cells in zebrafish. Huge amounts of pioneering rays can be identified in both ovary and the testes, and some of them can be mapped onto the T regions. We want to know if PV and pioneering pathway is still effective during ZTA. We analyze a small RNA data from 1 cell to 48 hours post-fertilization. We map the rays onto genome and collect the mapped rays from the active T loci. We observe a very clear pattern, which shows gradual decrease from 1 cell to 48 hours. Interestingly, we observe an increase from the belong to 40% apibody stage, which is overlapped with zygotic T activation. We think these rays may come from the degradation of T along transcript by PV and pioneering pathway. We also test the expression of PVL1, LL2, and other related genes, and find they can express during the ZTA, indirectly suggesting that PV and pioneering pathway may regulate ZTA. Although previous studies suggest that small RNA which can map onto T regions are pRNA, we want to provide more evidence to support it. First, these small RNA rays ranging from 23 to 33 nucleotide are consistent with the typical lengths of pRNAs. Second, 56 to 82% of small RNA rays can be annotated as known zebrafish pRNAs. We also investigate the sequence feature of this small RNA and identify an overrepresentation of uridine at the 5' most end and adenine at the 10th position, which are also another unique features of pRNAs. Taken together, our findings suggest that most mapped rates on activity along loci belong to pRNA species. A short summary to this section. ZTA can be regulated by chromatin accessibility and H3K9 trimethylation. Perhaps the PV-pRNA pathway may add another layer of post-transcription regulation on ZTA. For the T-alone transcripts exported into cytoplasm, there is another way to silence them and prevent T transposition. Our long-read RNA-seq data further improved the transcriptome in zebrafish and identified 1,079 novel T gene and 595 gene loci. Finally, we did some functional analysis on 2 of them. To better understanding the biological functions of these novel genes, we used WGCNA to identify gene modules with highly correlated co-expression patterns. Then, we did a functional enrichment analysis and found M1 and M2 modules have important biological functions relating to the embryonic development. According to the expression pattern, domain analysis, and the literature searching, we select a novel T gene locker CYT1 and a novel regular gene D2 for further downstream experimental studies. Let's take a look at CYT1 first. So this one have 4 T gene chimera isoforms with the DNA transposon inserted in the last exon. Transcript abundance of CYT1 can be detected in the fertilized egg and one cell stages and decreased until the shell stage. Therefore, CYT1 is likely a maternal gene. BMB gene, which is essential for mediating nuclear envelope fusion, is the only homolog of CYT1 in zebrafish. To obtain a knockout mutant of CYT1 gene, we employed the CRISPR-Cas9 system to modify it by targeting the first exon. The Zygotic ZIT1 CRISPR-Cas9 knockout mutant showed normal morphology during embryogenesis. Well, both the maternal and the maternal zygotic ZIT1 mutant fail to undergo cell movement during shell stage and cannot develop further. Therefore, ZIT1 is a strictly maternal effective gene and essential for early development. ZIT2 is a novel gene from the co-expression module M2, where the gene has specifically high abundance at 1K cell stage. This module is functionally enriched in regulation of cell cycle and translation. ZIT2 is a homolog of FASTK5 genes in human and mouse, which may have function involved in mitochondrial RNA processing. The zygotic CRISPR-Cas9 knockout mutant of ZIT2 develop normal until 6 days post-fertilization. After that, the fish show a body curvature phenotype and the apparent phenomena of cell death. Finally, all the mutants cannot survive beyond 18 days post-fertilization, and therefore fail to develop into fertile adults. Collectively, ZIT2 is also an essential novel gene for zebrafish early development. Okay, finally, I want to say there are lots of people that offer general helps to me on this project. First, I want to thank my collaborators, Dr. Dingjie Wang and Dr. Ting Li. They are also the co-first author on this manuscript. And I also want to thank Professor Shunji Jia and Yungui Yang and Yang Ying for their intensive discussion on this project. Finally, I would like to thank my PI, Dr. Qingfei O, for his instruction through the entire project, and he spent plenty of time to discuss the many details of this work and help me to interpret the results and make a lot of efforts in writing the manuscript. Thank you all. Okay, that's all for today. Thanks for your time.